Okay, so Hada, just introduce yourself in a brief.
Yes, sir. My name is Shuhada Begum. I am from Hojai district of Assam. My schooling and graduation was done in Hojai itself. To do my MSc, I moved in Guwahati, where I did my MSc in biotechnology from Guwahati University. After that, I have joined PhD in Tezpur University in 2021.
Okay, so Hada, so currently you are a PhD scholar, right?
Yes, sir.
What is the topic of your PhD?
My PhD topic tentative title is creation of Senior Solanacearum pathogen mutant and study of its virulence in tomato seedlings.
Okay, that means your area of that is plant microbe interactions, right?
Yes, sir.
Okay, so Hada, for the microbial interaction to the plant rule, that is the plant microbe interactions,
Yes,
there
sir.
are certain phenomena happens in the rhizosphere zone.
Yes, sir.
Okay, so can you elaborate how bacterial populations get attracted to the root region of the plant?
Um, As we know that this plant's root area is highly rich in microbes as plants secretes some alkaloids or other plant roots exudates its secondary metabolites and all which attracts the microbes present in the soil and through wounds and external cuts or and all microbe used to enter into the plants.
Okay, so that means bacterial has a also sensory organs to sense the
Yes,
these root exudates.
yes,
How
sir.
bacterial populations know that is there are some luxurious secondary metabolites secreted by the plants? How bacterial sense?
Bacteria have specialized sensory system. I'm unable to recollect the name to something kinase. Uh, Sorry, sir, I'm unable to recollect, but they sense the external environmental factor and they activate their signaling cascade through which they get again attracted towards the host plant or uh, organism.
Okay, Suhada, have you heard about the plant growth promoting rhizobacteria PGPR?
Yes, sir.
So what is this?
Those are the bacteria which resides throughout the rhizosphere of the plant, and they helps the plants and plant secretion helps them, and directly or indirectly, they also helps the plants growth and development.
Okay, so based on
For
your answer, that
example,
is there are two method of a PGPR to promote plant growth. That is one is the direct method and one is the direct indirect method, right?
yes, sir.
So
Yes,
what
sir.
are the possible way of a PGPR to help plant growth and development directly and indirectly?
Indirectly, we can say that in soil there are lots of inorganic substances which are not directly uh, taken by the plant, which has to be broken down and which is done by those PGPR microbes. And in case of direct method, direct process where those microbes enter into the plants and they forms some kind of structure. In case of leguminous plants, we can take the example of rhizobium. So they create some nodule formation and where they secretes uh, uh, synthesizes some kind of plant hormone. I'm unable to
Okay,
elaborate.
Suhada, have you heard about the exopolysaccharide EPS?
Yes, sir.
What is this? What is exopolysaccharide?
These are the secretions by the microorganisms. They forms film-like structure where through the exopolysaccharide secretion they adapt in drought condition. Uh, Also, they save themselves from external environment or external so if we apply antibiotics and those exopolysaccharide can inhibit the
What is the
aldehyde
role of these
receptor.
EPS?
This especially to protect or to adapt microbes in a adverse condition.
Okay, that means it is beneficial for the bacterial population.
Yes, sir.
Okay, suppose the plant root exudates materials will transport to the bacterial cells from the root spares, right? So suppose if there is a bacteria having a property of a producing EPS, that is exopolysaccharide, okay, so it will cause a barrier to the bacterial cell or outer cells. In that case, how bacterial cells will uptake the nutrient from the plant cells if there are exopolysaccharides?
So exopolysaccharides are not acting as insulator for the bacteria. They can absorb or uptake their nutrients from the suitable environments, and it depends whether they are wanting that particular substance or not, depending on which they will adapt.
Okay, so Hada, have you heard about the ACC deaminase enzyme?
So,
ACC deaminase.
sorry, sorry.
ACC, capital A, capital C, deaminase enzyme.
Sorry, sir, I have not heard about this.
Okay, have you idea about the hydrophore?
Yes, sir.
What is hydrophore?
So therefore is related to iron iron scavenging. They especially access iron scavenger for particular microbes.
Okay, in PGPR, certain group of bacteria has have ability to solubilize the phosphate, especially in organic phosphate, known as the phosphate solubilizing bacteria. Right?
Yes, sir. Yes, sir.
Why there is a need of a phosphate solubilizing bacteria if we have already chemical fertilizer like as a DAP?
Sir, in DAP, we are having in first compound form, which is not which cannot be uptaken by the plant if PGPR breaks it down into phosphate ion. Then only the plant can uptake accordingly.
Okay, so Hada, PGPR and GPCR, these two terms are very similar in sound. So, what is these two terms and how they differ from one another? GPCR and PGPR.
GPCR, sir, can you expand the GPCR?
Have you heard about the GPCR?
I think I heard about it, but I'm not able to recollect right now.
Okay, do you have any idea about the cell signaling?
Yes, sir.
So
Yes, sir.
probably you have heard about the GPCR.
GPCR, yes, yes, sir. This is cell signaling related. I was
G
right now only
G
focusing
protein
on
coupled receptors.
yes, sir. Yes, sir. Yes, sir. G protein coupled receptor, which is also known as 7 serpentine transmembrane protein.
Okay, so now you can just explore the differences in GPCR and PGPR.
Yes, sir. This is totally different thing. In PGPR, it is about microbes, and in GPCR, it's That's why I was unable to relate this to thing.
Okay, Suhada, there are three terms, that is autocrine, paracrine, endocrine.
Yes, sir.
What are these terms?
In case of autocrine, the cell secretes substances or hormones, and then those substances will act on cell secreting cell itself. In case of paracrine, it moves to a certain distance, then it will act on the distant cell. Uh, Also, in case of endocrine will it will act the secreting cell. It will act the cell far away from the secreting cells. The fourth term was which was the fourth
Autocrine,
term?
paracrine, endocrine.
This I have to find three of them.
Okay, Suhada, basically we know about the endocrine gland. There are so many glands we
Mm,
have and produces
yes,
different
sir.
kinds of hormones and have their own
Mm,
functions.
yes, sir.
Do we have also exocrine gland?
Um,
If yes, then kindly say some name of the exocrine gland.
uh, sir.
No idea. Okay, have you heard about the lacrimal gland?
Yes, sir.
What is this? What is lacrimal gland and where it is found?
Sir, I guess it is in our eye tears secreting gland,
Tears.
if I'm not wrong. Yes, sir.
Okay, okay, no issue, no issue. Just feel normal and comfortable.
Um,
Okay, Suhada, what is your favorite topic and subject?
Sir, my currently I cannot strongly say this is my favorite topic because I'm just I did my master's in 2019,
Okay,
so
no issue.
molecular biology and microbiology I can say.
Molecular biology and microbiology, right?
Yes, sir. Yes, sir.
Okay, very good. Uh, Swara, suppose you are a microbiology student and you are working in a laboratory. What kind of a precautions you have to take care of your laboratory as well as yourself in microbiology laboratory?
Uh, we have to maintain a sterilized condition where we have to save us as well as we have to save our sterilization is the main precaution has to be taken in microbiology laboratory.
Okay, in molecular biology, DNA has very crucial and important role. Okay, and the chemistry and the biochemistry and the metabolism for DNA is also very important. Okay,
Yes, sir.
suppose there are certain nucleotide like as ATP, ZTP, CTP, etc. What different kinds of chemical bonds are there in a nucleotide molecule?
Among nucleotides is phosphodiester bonds. Different types
What
of
is the chemical
protein.
composition of a nucleotide?
Nitrogenous base,
Yeah.
uh, phosphorus, uh, and sugar. This sugar and phosphorus is bonded with uh, glycosidic glycosidic. What is it?
What is the name of a chemical bond which join phosphate with the sugar and base with the sugar?
So I have to revise this thing.
Okay, no issue. What is nucleosome?
The nucleosome is uh, it is DNA wrapped around the. It is the unit where DNA is wrapped around the histone protein and uh, which is called which is in the form of octamer and which is termed as nucleosome.
Okay, Suhada, have you heard about the topoisomerase enzyme?
Yes, sir.
What will happen if there is no topoisomerase in the cells?
So in case of prokaryotes, if there is no topoisomerase, then it will disturbance will be created during uh, replication formation where topoisomerase creates relaxation in supercoil.
Okay, that means my question was what will happen if there is no topoisomerase activity? So, what will the consequences?
Uh, Replication fork will not form properly as topoisomerase does the proper relaxation during replication formation. It neutralizes the super negative and positive supercoiling in replication formation.
Okay, what is intron splicing?
It is after in case of eukaryotic transcription, we get the mRNA, which is with exons and introns, which come out with exons and introns. Introns are not non-functional; do not cause any kind of protein, so we have to remove those parts, and removing of those parts is called intron splicing.
Suhada, do bacterial cells also have such intron splicing process?
No, sir. As bacterial cells have their DNA in operon system, so they don't have any kind of introns.
Okay, operon. What is operon?
It is the cluster of gene located under a single promoter. One single promoter uh, can uh, can transcribe the more number of genes.
Okay, that means the cluster of a gene which regulate the function of a certain process. So, can
Yes,
you
sir.
name some operon in bacterial cells?
So, well-known operon is leg operon, then others are tryptophan operon, arabinose operon,
Acha, like operon or?
tryptophan operon.
Okay, very good. Uh, how many ATP molecules are required for the formation of a one peptide bond during protein synthesis?
Um, Sorry that I cannot remember.
Okay, RNA processing like as a capping, tailing, intron splicing. All this process
Yes, sir.
happens where? That is inside the cytoplasm or the nucleus.
So, capping is 3 prime capping is so inside the nucleus.
Inside the nucleus.
Okay.
Does bacteria have nucleus?
No, sir.
Then where it take place in the bacterial cells?
So there is a region where their genetic material resides. It's called
Do
nucleoid.
bacterial cells also go for the these RNA processing like as capping, tailing, and intern splicing?
No, sir.
Okay, okay. I I think I thought you were trying to say something. Okay, Suhada. Uh, in genetics, we generally uses qualitative and quantitative genetics, a term. What is the meaning
Yes,
of
sir.
qualitative and quantitative genetics?
Uh, in case of qualitative jobs, sir, I'm trying to just answer this out of my knowledge.
Okay, that
Um,
means you are not interested in the genetics.
Not much, sir.
Okay, okay, okay. So, in molecular biology,
Okay.
DNA polymerase has a very important role,
Yes, sir.
right?
Yes, sir.
And DNA polymerase has multiple functions, like as polymerizations, proofreading activities, and gap filling, etc.
Yes, sir.
Which activity is responsible for proofreading?
So it is 3 prime to 5 prime exonuclease activity.
Okay, and what about 5 prime to 3 prime exonuclease activity?
Um, it is gap filling, I guess.
Gap filling.
Okay.
Yeah. Okay. During DNA replication, during DNA replication, one strand DNA polymerase will move in the continuous manner, while another will move in the discontinuous manner.
Yes, sir.
Who will decide this?
Sir, DNA polymerase will decide this.
DNA polymerase itself decide it. How?
Yes, sir. Uh, because DNA polymerase as well as the um, direction of the strand. It is
In, in which
one moves
in
towards
which in which directions DNA polymerase will move?
5 prime to 3 prime.
Why? Why it cannot move in 3 prime
Because
to 5 prime?
in 5 prime and only it will get OH, free OH where it can add external nucleotide.
Okay, means DNA polymerase cannot move in the 3 prime to 5 prime directions,
Yes,
right? Only move in the 5 prime to 3 prime direction.
yes,
Then how it perform 3 prime to 5 prime exonuclease activity? Okay, okay, Suhada. Uh, uh, Now the term is over now. So now you are free and this session is open for you. If you have any query, questions, and doubt and suggestions, then you can just ask to me, and you can talk Hindi also. Now you have to feel normal and be relaxed. सो अगर आपको लगता है कि अगर आपको कुछ डाउट है, कुछ पूछना है और कुछ आपको सजेशंस चाहिए है, तो आप ज़रूर पूछिए। फिर इसके बाद आपको हम बताएंगे कि आपको किन किन चीज़ों को फोकस करने की ज़रूरत है और कहाँ पर आपकी लैक है। पहले आपका टर्न है, पहले आप पूछ लीजिए।
जी सर। सर मेरा प्रॉब्लम ये है कि मैं कंप्लीटली ब्लैंक हो जाती हूँ, मतलब क्वेश्चंस तो नॉन होता है जो कि मुझे आता है, ये मुझे पता भी है, बट whenever I try to attempt this question, मैं completely blank हो जाती हूँ।
सुहादा, ये जो प्रॉब्लम है कि एट द टाइम ऑफ इंटरव्यू या एट द टाइम ऑफ आंसरिंग आपका माइंड जो पूरी तरीके से ब्लैंक हो जा रहा है, उसके पीछे कारण ये है कि आप बहुत लॉन्ग टाइम से इन सारे कंटेंट को लेकर के बहुत दूर रही हैं। मतलब जो अभी आप पढ़ रही हैं एग्ज़ाम को लेकर के या जिसके लिए आप इंटरव्यू देने जा रही हैं, ये जो मतलब ये सारा जो कंटेंट एंड टॉपिक्स है ना, इससे इससे आपका काफ़ी गैप हो चुका है। आप उसके टच में नहीं हैं और आपने एग्ज़ाम के लिए उसको पढ़ाई किया हुआ था और एग्ज़ाम में आपने उसको लिखा था। इंटरव्यू के टाइम पे क्या होता है कि प्रेशर बहुत ज़्यादा होता है। आपके दिमाग पे प्रेशर होगा, दूसरा चीज़ तो ये भी रहेगा कि आपको सोचना है कि आपको बोलना क्या है। इन दोनों चीज़ों के प्रेशर में दिमाग आपका इस चीज़ को भूल जाता है कि आपको आता क्या है ना? उसके लिए
Yes, sir.
अभी तो फ़िलहाल यही सजेशंस है मेरा कि जितना ज़्यादा हो सके प्रैक्टिस कीजिए। आपका कब है? 12 तारीख को है इंटरव्यू,
यस सर, 12 तारीख
है ना?
11 है।
12 तारीख को है। तो कम से कम एटलीस्ट आप इतना कीजिए कि जो आपका इंट्रोडक्शंस का पोर्शन है उसको 4-6 बार रिपीट कीजिए। दूसरा, जैसे कि आपने बोला कि आपका मॉलिक्यूलर बायोलॉजी और इम्यूनोलॉजी या माइक्रोबायोलॉजी पे इंटरेस्ट है, तो उनके जो बेसिक कॉन्सेप्ट हैं, जो बेसिक टॉपिक्स हैं, उनको आप कवर कर लीजिए। अगर आप इम्यूनोलॉजी बोलती हैं तो आपको जैसे एंटीबॉडी है, एंटीजेंस है, लाइक एस, और आपका वैक्सीन है, राइट, हाइपर सेंसिटिविटी है, ये सारी चीज़ें आपको पता होनी चाहिए। और अगर आप मॉलिक्यूलर बायोलॉजी बोलती हैं कि आपका मॉलिक्यूलर बायोलॉजी फेवरेट है, तो डेफिनेटली आपको डीएनए रेप्लिकेशन, ट्रांसक्रिप्शन, ट्रांसलेशंस, ये, और मतलब पोस्ट ट्रांसलेशनल मॉडिफिकेशंस, एटलीस्ट इतनी चीज़ें तो आपको आनी चाहिए, म्यूटेशंस, और वो भी कंपैरेटिवली एनालिसिस पे कि ऐसा क्यों हो रहा है, ऐसा क्यों नहीं हो रहा है, ऐसा क्या है, क्यों नहीं है, इस तरीके से। तो इतना सब कुछ तो आप अभी नहीं कर पाएंगी, अभी आपके लिए प्रेशर ज़्यादा हो जाएगा। लेकिन जितना आप कर सकती हैं उस पर थोड़ा सा सब्जेक्ट पे अपनी पकड़ बनाइए, ठीक है? कोशिश कीजिए कि जो भी आपको आता है उसको आप बोलिए। बोलने में प्रॉब्लम है आपको, और बोलने के टाइम पर ऐसा लग रहा था कि जैसे आप एकदम नर्वस हो गई थी, और आपको ऐसा लग रहा कि जैसे आपके दिमाग में कुछ है ही नहीं, कुछ खाली हो गया एकदम से। दूसरा चीज़ ये है कि कई सारे आंसर्स आपके इनअप्रोप्रिएट लगे, मतलब कि जिनका कि ज़रूरत उस सवाल के लिए नहीं था, या जो सवाल मेरा था उसको लेकर के आंसर्स जो थोड़े थे वो करेक्ट नहीं थे। तो कोशिश आप ये कीजिए कि जब आप आंसर्स करें तो आंसर्स के टाइम पे आप वही चीज़ बोलें जो कि सवाल में डिमांड किया गया है, ठीक है? जैसे मैंने आपसे कई सारे सवाल पूछे यहाँ पे, फॉर एग्जांपल मैंने आपसे पूछा था कि न्यूक्लियोसोम मॉडल क्या है, या न्यूक्लियोसोम क्या है, या फिर मैंने आपसे पूछा था कि डीएनए रेप्लिकेशंस में आपका एक स्टैंड जो है वो कॉन्टिन्यूअस हो रहा है, एक स्टैंड आपका डिसकॉन्टिन्यूअस हो रहा है, ऐसा क्यों है? या फिर डीएनए पॉलीमरेज हमेशा 5 प्राइम टू 3 प्राइम पे ही मूव क्यों कर रहा है? इन सवालों के आंसर्स आपने थोड़े मतलब सटीक नहीं दिए, है ना? इसके पहले भी कई सारे सवाल मैंने आपसे पूछे थे। तो कहना ये है मेरा कि जब आप आंसर्स कर रहे हैं, तो आंसर्स के टाइम पे आप उतना ही बोलिए, और वो बोलिए जो कि उस क्वेश्चंस के लिए क्या हो, रिलेवेंट हो, है ना? बाकी की चीज़ों को बोलने की आपको ज़रूरत नहीं है, ओके? और तीसरा चीज़ ये है सुहादा की आपको बोलने की प्रैक्टिस थोड़ी कम है। फ्रिक्वेंटली शायद आप
यस।
मेरे ख्याल से बोलती नहीं हैं। तो उसको अभी ये 2 दिन का जो टाइम बचा हुआ है, 10 का, 11 का, 12 को तो आपको मतलब है ही वहाँ पे। 1 या 2 दिन का जो टाइम बचा हुआ है, इसमें आप कोशिश कीजिए कि जितना ज़्यादा हो सके आप लाउड साउंड में थोड़ा बोलिए। जब आप बोलेंगे ना तो आपके जो टंग होंगे, टंग टंग की ट्विस्टिंग होगी, और उससे आपको थोड़ा कॉन्फिडेंस मिलेगा, है ना? तो आपको 2 काम करना है। पहला ये कि जो आपका सब्जेक्ट है उस पे थोड़ी-सी आपको पकड़ बनानी है, उसको आपको पढ़ना है। सारा नहीं, कुछ-कुछ इम्पोर्टेंट चीज़ें, जैसे जैसे मैंने आपको भी बताया है। ये वीडियो आपको मिलेगा, आप उसको देख लीजिएगा। तो आपको वो करना रहेगा, और दूसरा आपको बोलने की प्रैक्टिस करनी है। ये 2 काम आपको करना है, ठीक है? ये 2 काम आप करेंगे तो डेफ़िनिटली आपको बहुत इम्प्रूवमेंट मिलेगा। सुहाना, ये बताइए कि अभी आपको कैसा फ़ील हो रहा है? देने से पहले आपका और अभी देने के बाद कैसा फ़ील हो रहा है?
वही नर्वसनेस था। अभी आपसे मतलब सजेशंस मिला है और मुझे क्या क्या मतलब इम्प्रूव करना है
बिल्कुल।
मतलब।
आप ऐसे ही अगर 2-4 बार और भी बोल के किसी को सुना देंगे ना, तो आपकी जो हेजिटेशन है वो दूर हो जाएगी, है ना? तो थोड़ा-सा कोशिश आप यही कीजिए, जैसे अभी सपोज़ कि मैंने आपका एक बार मॉक इंटरव्यू लिया, आपके मन में देने से पहले का जो कॉन्फिडेंस था और अभी में कॉन्फिडेंस से बिल्डअप हुआ होगा, है ना? आपको लग रहा होगा कि अच्छा, हमको
यस।
ये हमको ये चीज़ पे काम करने की ज़रूरत है। तो ऐसे ही आप अगर
यस।
2-3 बार अगर अपने आप को बोल करके किसी को सुना देंगी, तो वो आपके लिए ठीक रहेगा। ठीक है सुहादा? बाकी आप ग्रुप की मेंबर हैं, और आप के पास नंबर भी है। अगर आपको कुछ डाउट है, कुछ प्रॉब्लम है, तो आप ज़रूर उसको मैसेज करके पूछ सकते हैं। ठीक है?
यस।
तो इन 2 चीज़ों पे आप अच्छे से काम कीजिए, और बढ़िया से इंटरव्यू दे करके आइए। हमारी तरफ से आपको ढेर सारी शुभकामनाएँ और बेस्ट विशेस आपके इंटरव्यू के लिए। और
थैंक
दे
यू।
करके आइए तो ज़रूर बताइए कि कैसा रहा आपका इंटरव्यू, ठीक है? चलिए,
ओके सर,
कुछ
थैंक यू सर।
कुछ पूछना
थैंक
है
यू सर।
सुहादा?
थैंक यू सो मच सर, बस यही बोलना है कि आपकी वजह से
अच्छा,
मतलब
ओके।
इतना है।
मोस्ट वेलकम, मोस्ट वेलकम। ठीक है।
Okay.